Thank you, everybody, for coming to our presentation today entitled De-Risk Therapeutic Antibody Drug Development Using Humanized FcRn Mice. So if any of you there are developing antibodies or drugs, you're going to get to a point where you're going to need to evaluate half-life. And one of the riskiest things about evaluating half-life is not knowing the limitations of the assays that you've chosen. So I'll go through some of those and some of those platforms in the middle of the talk. To start off, I would like to introduce you to who we are in case you don't know who the Jackson Laboratory is. Our mission is to discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. And we do that in three ways, and this webinar is actually a nice marriage of all of those things. We have researchers in our Bar Harbor and our Connecticut facilities who are studying genetics and genomics and trying to develop therapies for human disease, just as many of you are. And in particular, we'll be focusing on the work of Dr. Derry Rupenian, who is a basic science researcher interested in autoimmunity, but has also very interesting translational goals in using that information about basic immunity to improve therapeutic responses. We also are a resource for mice and mice-related services to the biomedical community. And so we'll focus on a few of those mouse models. We have 11,000 mouse models, but today we'll focus on just a handful of those. And I'll be showing you some data that's coming from our services team who run therapeutic antibody evaluation services for half-life determination of new therapeutics. And I'll show you what we've validated on that platform that was developed by Dr. Rupenian's lab. And then, of course, we tie everything together in education. Our mission, especially in our technical information scientist group, is about educating scientists and improving their ability to do science and make discoveries better by the education that we provide. As you know, we are a repository for 11,000 mouse strains, as I mentioned before. We are funded through the National Institutes of Health to do so and to make those mouse strains available to researchers around the world. Importantly, we focus as a professional mouse repository on maintaining the genetic quality and animal health. And I think as a researcher, a bench researcher, I didn't really grasp the importance of these things. I definitely took them for granted. But these are really the basis for overall reproducibility in your results. And it's the use of the combined inbred strains that you can see listed here that you might be able to get from other vendors, at least different substrains of those mouse strains that are listed here. But it's those research dollars that you spend on our specific substrains that help develop the collection of our unique strains that cannot be found anywhere else, as well as our valuable, freely available resources. As I mentioned before, I'm going to be focusing on our in vivo services for therapeutic antibody evaluation and half-life determination, but we have a number of other services that would be related. So anybody who is developing a therapeutic would be probably developing them for some disease application. And so we've listed some of these that we specialize in. Primarily, I'll be focusing on oncology, but of course, there are many other places in which therapeutic antibodies are going to be used clinically. So the data that I'm going to be showing is coming from validation data that we've collected in our laboratory in Bar Harbor. And then we have several online resources to help you with your research. I'll leave these links here for you that you can use when you download the slides later, but I'll skip those for today in interest of the content. So the goals for this presentation are twofold, really just to introduce you to the models and why we think these models are important for de-risking your drug development pipeline in terms of understanding appropriate half-life, and how to choose those and how to use them appropriately in your research. And then I'll give you some examples of those types of studies using that platform. So to get to those two goals, I'll review a really important molecule called FcRn, or the neonatal Fc receptor, and what impact it has specifically on IgG molecules. And I will also talk about albumin, because albumin is another molecule that enjoys increased half-life by interaction with FcRn. Importantly, we'll focus on the differences in the ligand and receptor binding that occurs between different species, primarily human and mouse, and then provide to you our solution for humanizing the FcRn mice to improve your IgG lead selection and to establish appropriate dosing regimens. And I'll be going through some examples that are known in the literature, as well as data that we've collected in-house. So to get you started, I have these questions that I like to ask so I can better understand who's in my audience. And you'll see that over on the right-hand side of your screen. And I'd just like to get an idea of what type of therapeutic you are developing. And you can select over there on this side of your screen any of the options. And then also, if you move a little bit further down, you have to actually submit those answers so that we can see them. So I'm happy to learn about how I can approach the next 45 minutes by kind of understanding who's in the audience, and I can gear the presentation appropriately for that. So go ahead and put that information in on the side, and we'll give you just a quick second to do that, and we'll have another question coming up afterwards, and Kevin will display those results so I can see them. Great. So it looks like the majority of you are doing IgGs, and any of you that are also looking at Fc fusions, that's great. I see a few albumins. There's a couple of people who are doing other. I might be curious to know what you guys are doing later, because I do want to be able to figure out if I'm going to be if my talk is going to be applicable. And I also love seeing that people are here just for education. So that's great. Those of you that are answered A, B, and C, these are definitely going to be models that are going to be applicable to your research. Okay. Quick next question. What type of development, or what phase of development are you currently in? Are you very early on in the discovery process? Are you somewhere in the middle, lead validation, optimization, preclinical testing? Are you going into toxicity, or are you actually in clinical trials? And again, those of you that answered just here for educational purposes, I love that you guys are here. All right, and let's see what those results are. Great. So I see a good variety. There's a peak here in preclinical testing, so that's great. I think this would be a good time for you guys to come to this webinar and learn more about these mouse models. All right. So using your results in that chart that I had before, the use of mouse models is going to be relevant in many of these phases of development. Of course, probably not phase 1 as much. So the use of mouse models, particularly in these two phases where you're looking at your lead and trying to validate and optimize them, as well as test them preclinically for efficacy, are really going to be using a lot of mouse models, and potentially even humanized mouse models that we're discussing. So this is really where I'd like to focus our discussion, is really on efficacy, but primarily on half-life, and being able to choose the best variant of your antibody to determine concentration and frequency of dosing. So we'll come back to efficacy a little bit, and how that the model that you choose for your half-life determination might also allow you to also test efficacy, depending on what your question is. So I'll start with some definitions. I'm not a pharmacologist by any stretch of the imagination, so these are my versions of these definitions. And so for any of you out there who have far more training than I do, I apologize if I'm glossing over or simplifying. So for the purposes of today's discussion, I'll talk about pharmacokinetics, or PK, as a study of how the organism affects the therapeutic molecule, and it's really understanding the fate of the molecule from administration until elimination. And we're basically talking about the stability of that molecule in the blood and whether it's going to be able to affect the patient by just being there. The half half-life is the time that's required for the concentration of that substance in the body to decrease by half, and maybe that's being eliminated through kidney or liver processing, and it really allows us to determine initial dosing. I'll talk about monoclonal antibodies and IgGs kind of interchangeably, but this is the major component of humoral immunity, which links the antigen recognition, or that specificity, to an effector mechanism of the immune system. So what happens to that once the antigen is recognized? I mentioned albumin, which is a main serum protein that maintains osmotic pressure and pH. Very important to do that, but it also can be used experimentally for developing drugs. And then importantly, FcRn, which is the neonatal Fc receptor that maintains IgG and albumin homeostasis. It's primarily expressed in the endothelium, some immune cells in the adult gut, and some other tissues as well. And I'll be referring to them as H for human FcRn and M for mouse FcRn. The last term is transgenic, which you'll see represented as TG, and this refers to a mouse that contains stably inherited DNA that has been inserted randomly into the genome. And I bring that up because we have some shorthand notations in the slides that use the word TG, and it really refers to the mouse models that we're going to be describing. So it probably comes as no surprise to most of you in the audience who are looking at IgGs that monoclonal antibodies are pretty important in therapy. A lot of these molecules have been used for cancer, so I will focus on cancer for my examples, but there's definitely been use for autoimmunity and neurological diseases. So there's a lot of different applications where a monoclonal antibody therapy would be relevant. So some of the mechanisms shown here, why have we chosen monoclonal antibodies, and what do they do? So in the case of these tumor-specific IgGs, or maybe disease-specific IgGs to be more general, there's usually some sort of specific biological target or antigen that you have in mind where a protein is being overexpressed in the diseased versus the normal tissue. And so that antigen binding can be used to deliver drugs or block protein functions or deplete factors. So in the case of a tumor-specific antigen, there might be an upregulation of this antigen on the tumor, and the antibody can then come in and recognize those specific antigens and then bring effector cells or immune cells in to directly kill that tumor. There's a blocking function that's summarized here in B and C, where those antibodies can bind to receptors and then block the interaction of pathways that are important for the tumor to survive or to grow. And then a third major approach is to use antibodies to deliver specific types of payload, either in the form of antibody-drug conjugates or maybe radioimmunotherapy. And then this last kind of function here are some modifications where small pieces of those monoclonal antibodies are used to determine specificity, but then they may be linked up to other molecules that have different effector functions in the case of bispecific antibody therapy in CAR T cells. So for the majority of the molecules on this page, along the top and along the left-hand side, these molecules will enjoy the benefits of higher half-life stability due to their interaction with FcRn. And these molecules here, which do not have the Fc portion represented in their therapy, will not be able to benefit from this type of half-life improvement. So that brings me to why IgGs are typically used for therapy, and for those of you who are using IgGs or Fc or albumin, it's all using the same scientific principle. So specifically for IgGs, they actually have very high bioavailability and meet a lot of the tissues in the body, and so they're available to reach the targets of choice. And of course, they have the high target specificity that we've been describing. But compared to other classes of immunoglobulins, they actually have the longest half-life of about 20 days. So the benefits are less frequent dosing, greater efficacy, and increased patient compliance, and overall lower costs associated with being able to keep these drugs in the body longer. So what is it that's allowing the IgGs to be maintained in the body? Of course, it's our star FcRn. So this is data that's taken from Dr. Rapenian's laboratory at the Jackson Laboratory, where they're really interested in understanding what the heavy chain of this molecule was doing. And so they made a knockout of FcGRT, which is the heavy chain of FcRn. And you can see here in female and male mice, they assayed where they looked at the serum levels of the immunoglobulins in both the knockout in white and the gray wild type bars. So you can appreciate here that in the knockout situation that all the IgG subclasses are no longer being protected and they're leaving the serum. Interestingly, though, this is specific to IgGs because IgMs and IgAs were not affected. So the conclusion was that this molecule is pretty important for protecting IgGs. So really what's happening here, and this is just a monocyte or an endothelial cell for an example. So when proteins are floating around in the serum or in the blood, the endothelial cells are constantly taking them up through endocytosis. And they come into an acidified endosome and then are usually transported down to the lysosome and then degraded. What's special about IgG is that FcRn is expressed here, and FcRn and IgG actually bind at acidic pHs. And so these IgGs are rescued from this degradation pathway and actually are recycled back to the plasma membrane, where they can be released into the bloodstream at physiological pH. So the binding at physiological neutral pH is released, and then the binding at lower acidified pH is much stronger. So there are other factors that can alter IgG half-life aside from this biological recycling pathway. There are species and chimera differences that will be an important focus of the talk. So most antibodies that are being used in people now have a greater percentage of a human component in them. But the very first monoclonal antibodies that were used in people were actually 100% mouse, and they had a terrible half-life. And so we'll describe why that is in a few slides. A lot of antibodies may also cause immunogenicity, and so the antibody itself is actually causing an immune reaction from the host. And so the host then reacts and removes those antibodies from the system. And then, of course, there are a lot of biological post-translational modifications that occur, or even the chemistry that your chemists are doing to modify the stability can affect the half-life. And of course, if you're doing something like attaching a payload to this antibody, that can cause a lot of steric hindrance and also affect whether the molecule stays in the blood or not. So once you're developing an antibody, there's a lot of reasons why you need to perform both in vitro and in vivo half-life determinations. And you'll be using these techniques in many different combinations at different points in your development cascade. So I kind of break those into two big categories, either being in vitro or in vivo. And most of the in vitro models, for example, BioCore or surface plasma resonance imaging, relies on the ability to calculate an on rate and an off rate of a binding between an analyte and a ligand. So in the case of SPR, or surface plasma resonance, there is going to be some sort of molecule that is going to be affixed to a surface, and then there's going to be a molecule that can be flowed past at a certain rate, at a certain concentration, at a certain pH. And then that will, the sensorgram in this case, the reflection or the amount of light that is bouncing off of this surface, then determines whether the analyte and the ligand are bound or not bound. And that allows you to calculate on and off rates and determine the steady state. Importantly, what this does is allows you to make some guesses as to the affinity of the molecule, whether something is binding quickly or slowly, and at high or low concentrations. So the in vivo models, which are listed generally here, follow a really similar profile as each other or experimental paradigm as each other, in which you inject that molecule into the animal, and then you take blood samples over time and measure how much of the drug is left in the serum. And then you can take that information and put it into some software that will allow you to calculate the half-life based on what's been administered and what remains in the blood. So my next question for you is, what kind of methods are you using to determine half-life right now? Go ahead and answer. If you fall into kind of the in vitro category, this is kind of my catch-all for in vitro. Maybe you're doing surface plasma resonance, or maybe it's some cell culture-based binding assay. Or maybe you're in this kind of in vivo arena now, where you're using either wild-type mice, humanized FcRn, or non-human primates. Many of you will probably be using many of these methods at similar but different times of your development cascade. All right. And so I see those results right now. It looks like most of you are not currently evaluating, but you may need to in the future. So you're in a good place because you're coming for education to kind of learn what's out there. It's interesting also that some of you are using wild-type mice. I'd really like to talk to you guys in particular. You might be using it for different purposes, but I would say that for half-life determination, it's a really great time for me to talk to you about the limitations of using standard wild-type mice for half-life determination. So the rest of this chart is shown. You probably all know from experience, overall on the low end of cost are these in vitro assays, and on the very high end of cost are these non-human primates. And mice usually follow somewhere in the middle. Depending on your molecule, the ability to predict the human relevance can vary a lot in vitro, and you're probably all well aware of that. And in non-human primates, that correlation is usually very, very high. What's interesting, I'd like to point out to you, those of you who are not currently testing half-life and those of you who might be using standard mice for half-life determination, that, of course, these are very qualitative measures here, but there's a lot of variability in standardized mice. And I'll show you some data and some publications in which we show that the humanized FcRn mice would be a more improved model to determine half-life. So we'll start with the species specificity. So why do I care about you who are using wild-type mice for half-life determination? Well, it all comes down to binding. And so this is the SPR, again, surface plasma resonance, where the researchers here were comparing the species specificity differences between human and mouse FcRn and human and mouse IgG. So in this study, Ober and colleagues were affixing they were affixing the IgGs to the surface of the sensor, and then the FcRns were flowed over at a fixed rate and pH. So these were the bound, the immobilized IgGs, and then they were flowing the FcRn over. And so they did many different concentrations of these different FcRns, and A and B are representative plots from a fixed concentration for each of those FcRns. And then table 1 is a summary of that. So what you'll notice in A is that the curve is much higher when human IgG interacts with mouse FcRn than when human IgG interacts with human FcRn. So this difference right here. And interestingly, mouse IgG, when we look at mouse IgG, doesn't bind to human FcRn very well at all over here in B. So right here, this really just shows that the mouse FcRn binds to human IgG very tightly. So what this means is that the mouse FcRn is protecting human IgG better than human FcRn, and therefore when you're putting your IgGs into a mouse, a wild-type mouse, that mouse is not going to predict the appropriate half-life for that molecule because of the species differences. What I thought was really interesting here was that mouse IgG and human FcRn don't bind at all, and so this is interesting for the incidents when the very first therapeutic antibodies that were used in the clinic were of mouse origin, and we were putting them into humans. And of course, they weren't binding because they don't bind well, and it really can't explain why the clearance rate for those molecules was so high. So coming back to human IgGs, human FcRn should probably be used in place of mouse FcRn for better modeling. So those were the responses from Dr. Rapenian's lab. He took his knockouts, and his group actually made 2 different transgenic mice that expressed the human FcGRT, or the heavy chain of that FcRn molecule. One of them had the endogenous human promoter, and it's a huge back construct that was made. And then there was another construct that they made at the same time to see if a broad chicken beta-actin promoter would accomplish similar goals. And actually what they found out was they were glad that they did both, because the endogenous promoter actually provides the longest half-life of these humanized models and best predicts human half-life overall when it's compared to collected data and known data in human patients. And actually this other molecule, which seems like it may not be as accurate, is actually very good for discriminating subtle differences in PK among different antibody therapeutics. So if you've got a few molecules that you're trying to decide which one to move forward with, you might actually consider using this, what we call TG276 model, in order to pick the best lead amongst those candidates. So I'll show you how those compare side by side. So TG32, as I said, is the most accurate representation of the human condition, which I'll show you in a few slides. And then the TG32 is the one that allows you to see these subtle differences. So the graphs here are basically measuring how much of a molecule is remaining in the serum. And there were 2 different variants of these molecules, either variant 1 or variant 2, shown in the solid versus the open shapes. And then the mouse models were compared either in blue or in green. So the TG32 is in blue, and then the TG276 is in green. And so what I hope you notice, whether the mice are homozygous or hemizygous, the overall concept here is the same for both of them, in that the TG32 has the highest half-life, and that the TG276, actually you can see a difference between these 2 variants. So what's really interesting about these, even though this is the endogenous promoter, the chicken beta-actin promoter allows you to show this difference because the binding isn't as good. So it allows you to distinguish subtle differences in your variants. So that's data that was collected from the Jackson Laboratory in our Bar Harbor in vivo pharmacology services group that does our therapeutic antibody evaluation services. But this is also shown in literature, and there's a lot of examples in the literature, but I'll just focus on this one early publication in 2014, where they looked at 11 different molecules that were approved to be used in clinic and had human data associated with them. You'll notice that most of them were IgG, but there are some Fc fusions. So they did a correlation study taking known data from humans and non-human primates, and then tested the PK using, in this case, the TG276 model, as well as wild-type mice. So the correlation to humans is shown here, and they use TGM as their abbreviation for the transgenic mouse TG276 model, FcRn model that we're talking about here. They also compared the wild-type mice here and then the known monkey data. And so if you look at the R correlation values, you can actually see that in some cases, the mouse transgenic predicted equally or slightly better than the monkey data, and that the data in the wild-type mice was a lot more variable. We also did studies like that as well more recently, and this was actually presented at AACR last year by Greg Christensen, who's our study director for our therapeutic antibody evaluation services. So he was using really clinically relevant PD-1 inhibitor or anti-PD-1 molecule pembrolizumab and doing a PK study very similar to what I showed you in the previous slide. Just again, comparing TG32, which is the molecule that has the usually has the best correlation to human data, and then the TG276, again, with two different genotypes of the mice. This is the one that allows you to distinguish different variants of your molecule. And so, of course, we don't have a variant of our molecule here, but just wanted to validate that this platform was relevant, could be used for clinically relevant molecules that are being tested and recently relevant. So you'll notice here, summarized as expected, the knockout mouse has really poor retention, and the wild-type mouse does have retention and binding. We also they also tested apilimumab, which is an anti-CTLA factor, anti-CTLA-4 antibody, and also measured those same types of results. Again, the FcRn being a nice control, showing that the binding is important for maintaining stability in the blood. And then the last one was belatacept, which is an Fc fusion, and so you can see some of that binding data here. So I went through those kind of quickly because it's all summarized in this next slide here. But basically, if we put belatacept, apilimumab, and pembrolizumab along the x-axis, and then the half-life determination along the y-axis, and then plot them based on the reference of human half-life and the half-life that was calculated in these different mouse, wild-type mice, either wild-type mice in gray, or these FcRn mice in blue and green, or the FcRn knockout, you can see that overall the Black 6 actually has the most variability out of all of them. So the humanized FcRn still follow this nice kind of correlation similar to what's being seen in the human, but these wild-type mice are kind of unpredictable. And that's actually shown here in the correlation data as well. And you can focus on the half-life of the TG32 homozygous mice here in comparison to human data with an R-squared value of 96.8%. And that is compared or contrasted to the half-life in wild-type Black 6 mice versus human, where that R-squared value is less than 50%. So I hope I've convinced you with this data and this comparison to the human data that the FcRn molecules are far improved over wild-type mice to determine half-life. So if I can test your knowledge now on my fourth question, what are some of the limitations of using wild-type mice in the humanized IgG PK studies? Go ahead and answer on the right-hand side of your screen. All right, do mice bind human with low affinity? Do the mouse FcRn bind with high affinity? Are wild-type mice less expensive than humanized mice? There are no limitations. All right, looks like you guys would answer pretty quickly. Let's see what those results are. Awesome. Thank you all for following along. So most of you said that mouse FcRn binds human IgG with very high affinity, at least from those binding assays. And based on the data that we showed here, that doesn't always translate to a predictable response of how it's going to look in the human. It's actually very variable. I think there's a lot more. Those surface plasma resonance data is, again, in vitro. Once those molecules get in vivo, clearly there's a lot of other factors at stake that are changing that half-life determination. All right, so let's move on to a related topic. There is the formation of anti-drug antibodies, or in the case of this paper, it was anti-therapeutic antibodies. So I mentioned that this was something that can cause differences or alterations in half-life, and that's basically when the host is seeing this therapeutic antibody as foreign and is trying to remove it from the blood. And so when you are doing these in vivo PK studies, you might see this as initially seeing the presence of the antibody or the molecule in the blood, and then suddenly seeing this big sharp decline in serum levels. And so this type of pattern is usually attributed to anti-drug antibodies. So some ways you can manage that, you might have seen it in the slides that I showed you, our validation data, that we excluded some of these affected animals. So once we saw that kind of drop, we took those mice out of consideration for that analysis, and that allowed us to then still calculate a half-life using the linear data. You can also potentially use the linear data prior to the onset of that ADA, but if it's not long enough, you may not have that choice. But the third solution to this is actually using the immunodeficient mouse strains, which won't have the ability to mount an immune response to the therapeutic. So of course, this is not going to be something that is going to help you in most cases in humans, but it does help you answer the question at the time. So we do have variants of these models. So these, again, just for your nomenclature understanding, FcGRT is that knockout of the mouse FcRn, and then if you see a SCID mutation or a RAG mutation or an NSG or a NOD SCID gamma, these are all different variations on how to make immunodeficiency in mice. The SCID and the RAG mutations that are listed here are important for B and T cell development. So these mice lack T and B cells, and therefore they don't have the ability to make their own antibodies. And then, of course, there's the different variants, the TG32 versus the TG276. So I'll show you the data on that. It's pretty short, but importantly, we did some validation showing that the presence of these SCID or RAG mutations or putting them on the NSG background actually, modify that. The SCID mutation did not affect the PK compared to the mice of the same genetic background, of the same transgene, but were not immunodeficient. So the presence of that SCID mutation did not affect the predictability of the PK or the PK to specific molecules like Enbrel, for example. So it's shown here in both cases. So the benefit of this is it avoids anti-drug antibodies. So if you have a molecule that you think might elicit one of these types of reactions, it might be worth considering using these immunodeficient strains. Also, the benefit of showing you this data is that if, for example, you started using one of the molecules that was not immunodeficient and then realized that you had an ADA, you could then switch over to the immunodeficient strain for your further studies without having to reproduce the original studies because the data was comparable. And then I mentioned the NSG variants. This will allow you, if you're thinking far in advance, you might actually be able to use these for your efficacy studies and be able to transplant human tissues onto them at the same time and perform efficacy studies that have your molecule in a more realistic, with a realistic dosing and more realistic half-life. I'm now going to switch gear for those handful of people who are going to be developing albumin conjugated molecules. And again, everything that I said related to IgG also applies quite well to Fc conjugated molecules as well. I think the ADA might be a bit different in those Fc fusion proteins versus IgGs, and so that might be a consideration for you. But for those of you who are also using albumin to conjugate your molecules, albumin also binds FcRn. It actually binds it in a different location. So IgG and albumin both bind FcRn, but they do not have to compete for the same binding locations. So very similar data as before, but now slightly confusing. The colors are reversed. So the wild-type mice, FcRn wild-type mice are in white, and the knockout mice are now in gray. And the same type of study was done where they compared the knockout to the wild type and showed that albumin does indeed get protected through FcRn, and again, through a different site than the IgG. And this is specific to IgG and not to IgMs, or sorry, this is specific to albumin, excuse me. And again, that repeats that data from before, showing that it was for IgGs and then not applicable for the other molecules. And again, similar story here. So this is, again, surface plasma resonance analysis done at pH 6, which is the relevant binding pH. And again, that same theme of the species differences between humans and rodents comes into play when we're talking about FcRn and albumin. And interestingly, the binding in this paradigm is a little bit different. So when you do the same type of study, the binding actually suggests that human FcRn binds mouse albumin really tightly. So if you are starting to test all of your molecules in a humanized FcRn mouse, you also now have to contend with the mouse albumin competing for that binding. And so if you are in the situation where you're using an albumin conjugate, you actually also have to knock out mouse albumin. So the mouse albumin will outcompete the human albumin on human FcRn, and so we have to get rid of that and address it. So we do have those molecules or those mice in which the mouse albumin has been knocked out. The mouse FcRn has also been knocked out, and then the human FcRn has been replaced. And so right now we only have a TG32 version, but we're also we have the version also in which there's immunodeficiency as well. So that can allow you some other options for mouse models to test. So in this case, this was also tested in the side-by-side assays showing how this knockout of albumin helps improve the half-life of, in this case, human serum albumin. So the half-life here is very, very high when you have the human FcRn and an albumin knockout. And again, the implication, again, is also that the mouse FcRn is knocked out. All right, I'll summarize by saying there are a number of humanized FcRn mouse models to help you with your research. I've categorized them pretty broadly based on the type of promoter that they have. So TG32 has the endogenous promoter and has the longest half-life, and that's shown here as well, TG32, TG32. The TG276 models that are shown here allows you to distinguish these subtle differences in PK. Some of these mice are immunodeficient, which can address ADA, so anything that has an NSG on it or has a RAG or a SCID mutation would be considered immunodeficient. And then there are some molecules, again, that we use for albumin, which you'll see listed as albumin knockouts here. So hopefully you can take this chart in your mind and answer the next two poll questions I have, which are conceptual questions and may require a little bit of thought on your behalf. Let's imagine now that you're trying to choose a model and you have 5 different IgG variants and need to select maybe 1, maybe 2 variants with the longest half-life. Which of these molecules, or sorry, which of these mice might you choose to proceed with your study? And just to orient you, all of the mice on the top are black 6 genetic background. The mice on the bottom are NSG. I've organized them as TG32 versus TG276, either with a SCID mutation or an albumin knockout, and again, TG32, 276. And I know this one might take a little bit longer for you guys to think about, so I'll give you some time to think about that. Trying to pick the winner of 5 down to 1 or 2. Which of these models will help you distinguish subtle variations in half-life? Okay, so I see there's an equal distribution of answers, so maybe I wasn't able to be clear about my messaging, so we want to go back here. So we'll go back to this chart here. Longest half-life is using the TG32 molecule, which has the endogenous promoter. These are the ones that had the highest half-life. We saw the half-life values very high along the top. The TG276 models, they have that CAG promoter, non-endogenous promoter, different species. This transgene is not expressed in the same tissues as TG32. They don't actually bind as well. And actually, because they don't bind as well, this is a good thing because it allows you to distinguish small differences in PK. The TG32 molecules all bind so tightly and so well that it doesn't matter. It just eliminates any kind of differences between them. They just bind incredibly well. These 276 models actually allow you to see those small differences because it actually doesn't bind to your IgG as well. So you can actually see that spread and difference between different molecules. So the answer is anything with a TG276. So it would be B or F. All right, another chance. Let's see, last chance in this presentation to see what model might you consider if you were worried that your therapeutic might generate some sort of host or mouse immune response. And again, it's the same organization of answers listed here, different question, different application. But hopefully the concept is clear. And I'll find out in just a few seconds when I see your guys' answers. If you're worried about immune response, which of these mice might help reduce an immune response? All right, Kevin, I think you can close that out. I know it's kind of a tricky question, but let's see those results. Great. Okay, so I see that most of you guys chose something with a SCID mutation, so C, D, E, and F. If you're worried about some sort of antibody response from the mouse, mice that don't have T and B cells, specifically B cells, would be a way to go. So anything with a SCID mutation or the NOD-SCID gamma, which also, of course, has a SCID mutation. So great. All right. So I'll summarize by saying conventional mouse models may fail to accurately predict PK. There's a lot of variability in what they can do because of those differences between human IgG binding to either mouse or human FcRn. You can get clinically relevant data using these human IgG molecule half-life, or you can get clinically human IgG half-life predictions possible using these TG32 FcRn mouse models, and you can also distinguish between different categories or different candidate molecules using the TG276 humanized FcRn models. And of course, there's a lot of variations on that platform, so depending on what your molecule is, there's a lot of ability to kind of more selectively choose a model that works for the type of molecule you have. So I will acknowledge all of the people who helped me with this presentation. Of course, Dr. Deri Rupenian, who developed the models, and of course our Jackson Vivo Pharmacology Services group, who did all of the validation, who run the studies for you. In particular, I'd like to thank Greg Christensen, who is the study director and the person whose expertise I'm trying to emulate in this presentation, as he really is the expert in understanding kind of the intricacies of using these models, and he can really help you refine the model selection based on the type of molecule that you have. That group has run about 100 different studies using different types of molecules, either for internal purposes or client studies. And so they have a lot of ability to pick and choose based on their experience. So that will bring me to the last slide. As I said before, we have therapeutic drug evaluation services. But a lot of you may be interested to know we also do efficacy testing for patient-derived xenograft cancer models, humanized models, immuno-oncology models, where I think those NSG variants FcRn may become very useful in the future. But we also have a number of other services that we've listed here. So if you are interested in contacting us for further information on any of those, feel free to reach out to that MiceTech email address